Construction of a Vaccinia Rabies Glycoprotein Recombinant Vaccine and its efficacy. National Institute of Immunology, New Delhi, India. Every day we encounter numerous pathogens. Mostly our self-defense mechanisms protects us. Certain others we get actively immunized. Few other diseases which we don't commonly encounter we receive post-exposure vaccination. One such disease is rabies. In India, mostly after an animal bite policy remains post-exposure vaccination. The disease affects all warm-blooded animals. Rabies is a 100% fatal disease if the infection sets in. Unfortunately, unlike many diseases, there is no recovery. It had been a challenge to the modern society. Every year it costs nearly 50,000 lives globally, mainly in India and Africa. Besides the affected individual, it takes a toll on the families too. The disease was contained in US and Europe through Vaccinia Rabies Glycoprotein Recombinant Vaccine Rab oral distributed as oral baits. Please read the early development of the Vaccinia Rabies Recombinant Vaccine Rab oral, 2021, Twists and Turns, Failures and Success, good paper. Fortunately, we did not have such issues. There was a veterinarian in our group who had previously handled fowlpox and pigeonpox virus. He had also spent 2 years at Institute of Veterinary Preventive Medicine, IVPM, evaluating safety and potency of veterinary vaccines. We never had doubts about the vector or the procedure. Vaccinia pox virus was agreed upon for it settles 2 issues: expressing rabies glycoprotein gene and delivering it as a vaccine. Gene was provided by Saker. But at the time of development, we never knew of the functional ability of the gene and if it will work as a vaccine. Hope was the paper print sequence, computer translation, and everything else will be in place, and fortunately it did. We also made a cDNA lambda GT11 library with LEP rabies virus infected mouse brain mRNA samples and the sequence X69061, available in GenBank. Europe and US have reduced the incidence of rabies in raccoons and foxes, mostly by oral vaccination distributed as baits. Similar strategy can be adapted to reduce the incidences in India too. But the target population is different: unvaccinated stray dogs. A recombinant vaccine was developed in 1987 at the National Institute of Immunology, New Delhi, similar to Rab oral, and its effectiveness was demonstrated in mice. The vaccine and its efficacy was presented at the India-US Vaccine Action Program, New Delhi, Indian Veterinary Research Institute, Isatnagar, Indian National Science Academy, New Delhi, and at the Central Research Institute, Kasauli. Below is the description of the vaccine and a schematic diagram how to make a recombinant vaccine. Need of the situation with eradication in mind requires a cost-effective vaccine that's easy to produce with long-lasting immunity that could be easily administered in a single dose. Unlike the present-day vaccines, unfortunately requires multiple doses, which is not practical for an eradication strategy. PSC11 Vaccinia virus vector was used for making our recombinant vaccine in a single dose. The vector has 2 promoters opposite to one another. One promoter drives the expression bacterial enzyme beta-galactosidase gene that gives the blue coloration in the presence of SCAL, and the other promoter drives the expression of our desired protein, in this case it's rabies glycoprotein. Flanking the whole expression cassette are the thymidine kinase regions of Vaccinia virus, facilitating homologous recombination with the Vaccinia virus. important to note, at the time of development, back in 1986, there was no precedence of similar genetically engineered vaccine for rabies using a viral vector was available for reference. Most of the development, like where and how to vaccinate, how much of recombinant rabies virus to be used for vaccination, how long to wait after vaccination for immunity to develop for a recombinant vaccine before challenge, how much virus and how to challenge. Most of the assumptions were based on general knowledge of pox infections and experience with fowl pox and pigeon pox vaccines from IVPM and advice from other vaccine facilities and veterinary institutions. Sekar Chakraborty joined NII, New Delhi from NIH, USA, and we rapidly progressed to construct Vaccinia Rabies glycoprotein recombinant virus as shown in the schematic diagram. Co-expression of LAC-Z gene helped to identify the Vaccinia Rabies glycoprotein recombinant viral blue plaques were picked and amplified. Co-expression of LAC-Z gene helped to identify the Vaccinia Rabies glycoprotein recombinant viral. Blue plaques were picked and amplified. Expression of rabies glycoprotein was verified by tissue culture cells infected with recombinant virus and reacted with fluorescent isothiocyanate conjugated anti-rabies antibody. Experience from IVPM helped to evaluate safety and efficacy of the vaccine. Efficacy as a vaccine of the recombinant virus identified in the previous slide was verified by challenge. Studies in mice. Mice were vaccinated at the base of the tail by scarification after 14 days both. Vaccinated and controls were challenged intracerebrally with LEP strain of rabies virus. Vaccinated mice were active. Pock lesions can be observed at the base of the tail and control mice are slowly dying. The development was announced to the press. Many national newspapers, radio, and TV carried the news. I was told it was even discussed in the Indian Parliament. Few newspaper clippings are provided for reference. The study was part of PhD thesis. Copy can be obtained from librarian, National Institute of Immunology, or please write to chandraraji at aol.com, chandraraji@aol.com. Low egg passage rabies virus can be obtained from the director, IVPM, Ranipet, Tamil Nadu, India. This development makes it possible for a cheap and affordable rabies vaccine. Spread to the unintended and immunocompromised population may be of concern. But the vaccine is intended for animals, mainly to dogs. We have watched many YouTube videos from India and other places on rabies. To my knowledge, it's the first message showing the efficacy of the vaccine developed in India by a recombinant DNA technology. A similar vaccine Raboral VRG, oral rabies vaccine for wildlife, is being marketed by Boehringer Ingelheim. We have also cloned and sequenced the glycoprotein gene of low egg passage, LEP, rabies virus obtained from IVPM, Ranipet, India. Spread to unintended general population is of concern about Vaccinia virus, mainly to immunocompromised individuals. Virus carrier can be changed to non-replicating fowl pox or canary pox virus. These pox viruses, in addition to pigeon pox, are being used in the poultry industry and are host restricted and will not replicate in dogs and cats, but express the protein and provide immunity. This development provides a possibility of eradication of the disease. 4. Further information, please write to chandraraji at aol.com. Please mention, I in the subject line. Rabies. Glycoprotein gene. The Vaccinia virus had been well studied. Many additional developments remains. To make the vaccine easily accessible, it needs to be adapted to oral dosage format. This should be possible with collaborative efforts of the dog feed manufacturers. Expressed protein by itself poses no problems, but still needs to act as a vaccine. Changing the vector to non-replicating host-restricted fowlpox or canary pox virus yet immunogenic may solve the problem. We wish Department of Biotechnology along with NII can direct veterinary institutions or universities to take up further studies. We are very confident this vaccine, combined with sterilization of stray dogs, make control of rabies a possibility. Pox viruses in general have a large genome. As shown here, it has the capability of expressing multiple genes. Our vision, in addition to control rabies, is also to establish a facility for analytical chemistry and tissue culture. We feel IVPM may have the required space and expertise. We thank Google to have provided a forum to spread the message.